So our final speaker of this session will be Galen Wright from the University of Manitoba in Canada. He's an assistant professor and Canada Research Chair in neurogenomics, and his talk will be Genomic Modifiers in Clinical Practice and Therapy.
Hey, thanks everyone for sticking around right until before lunch. I appreciate it, and it's great to be part of this session. So, yeah, as was mentioned, I'm currently an assistant professor at the University of Manitoba, but much of this work that I'm presenting today was when I was a postdoc in Michael Hayden's lab. I subsequently moved to Manitoba in April 2020, right at the start of the pandemic. I wouldn't recommend starting your research group as COVID hit, but we're gaining traction, and yeah, cool. So before we start, why should we be looking at these genomic modifiers, particularly with HD, and why particularly in human cohorts? Well, what I see is the two main things that we can get from it. We can improve predictions for age of onset models, but we can also then expand to look at preventative strategies because we're going to expand the number of therapeutic targets that are available for us to look at. And in addition to that, there are other advantages, particularly with using human genetics to inform these studies. And one of them that with precision medicine is frequently cited is the fact that we know that drugs often fail in clinical trials due to lack of efficacy, and these are very costly. And when we go and look at clinical targets that actually passed, if we look at they had human genetic evidence supporting them, we can see that it actually doubles or more the success rates of drugs in the therapeutic pipeline. And as recently as last year, if you look at the newly approved drugs, 2/3 of them actually had human genetic support. Beyond that, human genomics lets us understand new things about genes, about diseases, about processes. And this is just a short comment that we published earlier this year, inspired by some of the earlier work that was presented today from the University of Milan, showing that Huntington itself has got these really important neurogenic roles. And we went to go on and ask, what is this genetic variance, mainly SNP-based, what is involved in human cohorts? So we went and we scanned a large number of human genome-wide association studies and found 10 associations which are fine mapped using machine learning, and it helps advise core HDT biology. Some of the interesting signals that we detected were involved in the non-cognitive aspects of educational attainment, but also with regards to longevity and aging-related traits. So that was just a quick highlight of that study. But let's move on to the HD modifiers, and a lot of it's been presented already this morning, but I was going to go and do some of the greatest hits that inform some of the studies that we've been looking at today, and it's by no means comprehensive, but it just highlights some of the most important ones. But I think HD is really an important disorder to help us inform genomic modifiers and Mendelian disorders because we have these large cohorts because of collaborations with affected individuals. As we all know, back in 1993, it was recognized that the HTT gene in the CAG repeat was responsible, the pathogenic mutation, and it's been shown numerous times this repeat age of onset is inversely correlated, but it works well at a population level, but not so well at the individuals. Relatively soon after this, it was appreciated that somatic instability seems to be important in the primarily disease-affected areas of the brain. So if you look at the putamen caudate, you see it has in HD individuals affected by HD, you see that instability is the highest in these regions, and other areas such as the cerebellum, it's not as great. This pattern was seen. As far back as 1995, there were studies that identified that the CAG repeat is a complex region, and there are these variants within these in certain individuals. The study found a mutation that we'll talk about a bit later, but it was appreciated more looking at somatic instability and the ability of this variant to affect diagnostic tests, but we didn't look at the effect of age of onset. So fast forward a whole like 20 years or so, And the big breakthroughs or the confirmations studies that came through were these genome-wide association studies that then identified DNA repair, and it was mentioned particularly mismatch repair in being important, involved in these residual genetic, the clinical onset of HD. And then 4 years later, there were these findings with regards to these interrupting sequence variants and uninterrupted CAG on age of onset, which was covered in HD Buzz. And back in 2019, there were these 3 concurrent studies that showed that CHE length or uninterrupted CHE length is more predictive than poly-Q length in predicting age of onset. So I'm going to focus on what we found in one of those studies, which was published in the American Journal of Human Genetics. And the study was a follow-up from one of our the Hayden Lab's previous genetic modifiers in HD. And we were aiming at identifying further cis modifiers. For a number of years, we've been sequencing this region using Sanger sequencing and clonal, using bacteria to accurately characterize the repeat tract. And before I go on, it's important to always consider what this tract looks like and how we look at this diagnostically. So if we look at the diagnostic repeat, we use the value that's determined by the boxed value, but downstream there are these 2 additional glutamine codons followed by this polymorphic proline repeat as well. When we were sequencing, we found this variant which was mentioned and discussed earlier today that extended the uninterrupted length of the CHE repeat, but not affecting the polyglutamine tract. And when we looked at our UBC HD cohort, we found 16 HD patients from 6 families. And interestingly, about 3/4 of them were in the reduced penetrance range where they were symptomatic. When we looked at model predictions for these individuals, we saw that they presented dramatically earlier based on diagnostic estimates, so in the full penetrance range around 13 years and reduced penetrance almost 3 decades. And we also, I'm not going to cover it today, we also we identified the duplication of interruption variant with the opposite effect. We were there really interested in characterizing the effect of this variant in a follow-up study that was published in 2020 in Genetics and Medicine. We went and combined the results from different cohorts from the American Journal study as well as the GEM-HD cohort, and then a new cohort to have almost 50 loss of interruption individuals. And we found that obviously that when we corrected for diagnostic size and looked at uninterrupted CAG, it was more accurate as shown as orange, but these patients are still presenting around 10 years earlier than expected. So there's something more probably going on. We also went to look in whole blood in a number of individuals with the loss of interruption variant. And we showed when we looked at this diagnostic CAG size, which underestimates uninterrupted CAG by 2 and loss of interruption carriers, we saw that they had increased somatic instability. However, when we corrected for this, we found that there was no difference in around 160 individuals. So this is different from what we are observing with the age of onset predictions. We've got some preliminary data that Haley Findley-Black, a PhD student in the Hayden Lab, has been working on, where we've been looking at whether we could look at somatic instability for these variants within brains of affected individuals. So we've got a small cohort from the cortex where we showed similar patterns to what we're seeing in blood. So it's a small sample and there may be technology limitations or the cell types may have degenerated, but we still can't rule this. It's a complex situation now at the moment. So what are the clinical implications of these interrupting sequence variants? In 2020, we published a short review in Lancet Neurology looking at some of these. And one of the main take-homes is current HD diagnostic tests do not take the length of this repeat. They don't look at the DNA sequence, and they just look at length. And if you're going to do that, then you're going to miss these interrupting sequence variants, which, even though they're rare in the general HD population, they're still important for certain individuals. And one of those classes are these symptomatic RP individuals. So in the UVC HD Biobank, we looked at every single symptomatic RP individual, and we showed that like around 3/4 of these individuals in the whole RP population carried loss of interruption repeats, and they're particularly important in the lower bounds of this repeat class, so CAGs 36 and 37. We also went on to develop a loss of interruption specific PCR assay, which can then genotype without using DNA sequencing. There are also numerous new avenues that these findings that the field has come across in recent years from genome-wide association studies and also from these interrupting sequence repeats. So one of the therapeutic implications, especially in the field of Huntington disease, we know that these DNA repair and repeat instabilities represent an alternative approach, and it's important to expand our toolkit when looking at drug targets because we know Huntington's an essential gene. And one of the things outside of Huntington disease, which is neat about this approach, is actually looking at the shared mechanism, which we can look at the other repeat-mediated disorders, so almost 50 of them, as well as CAG and CTG repeats. And what's the evidence for the overlap of risk? So over the summer, I had an undergrad student who was looking at a recent repeat expansion GWAS, which highlighted some of the mismatch repair genes and be important with age of onset. And all we did is we went and we plotted the top hits from the most recent GEM-HD GWAS, and we looked at the effect size in the HD cohort on the x-axis, and then in the XDP, repeat expansion disorder, in the y. And you can see there's a clear correlation. The R-squared is about 0.8. Even though the motif for the disorders are different, they're affecting in a similar way in these individuals or in these individual diseases. So what is currently being explored? So one thing that we could think of is we know that these interruptions are important. So if we can develop CRISPR-Cas9 to maybe add additional interruptions into this repeat. There's also the way to look at this where there's small molecules, Christopher Pearson's labs exploring some of these molecules that bind to these slipped out formations that could prevent somatic instability. And then finally, there are different approaches to looking at these DNA repair genes, and we can target them with small molecules or ASOs, and MSH3 has been the subject to the most research in this regard. I don't have time to go over it today, but in our rapid review, we listed a whole lot of different pros and cons and future challenges of these different approaches, which I encourage you to go over if you're interested in this. At this stage, and it's always nice in research to have more things to do, so then unanswered questions and future directions for these areas of research. So we also need to look at what the off-target and unintended consequences of targeting HD genetic modifiers. So this just shows pathogenic variants from ClinVar for the main GWAS HD genomic modifiers. And if we look in OMIM, we know that there are various types of cancers that are also associated with these disorders. As was also highlighted in in Darren's talk earlier today, there are these other variants, and like in certain populations where we're seeing that even outside of the polyglutamine tract, there are areas that are the downstream proline tract, so synonymous mutations there. And if we look at the loss of interruption variant, even though we're seeing that the individuals are presenting earlier, in our cohort, more than 50% of individuals actually carry this polyproline variant, which takes away the CCA codon. So that may be a potential reason why we're seeing an increase, a more significant reduction in age of onset in some loss of interruption carriers, and we should focus that for future research. And so definitely we need to start characterizing these modifiers and mechanisms, also looking at processes out of somatic instability and DNA repair. GWAS is teaching us that it's also this two-step mechanism where you have somatic instability with downstream, which then triggers downstream cellular death, and some of the modifier genes are important in that regard for the second step. And then also with the interrupting sequence variants, starting to look at what could also be involved in them outside of somatic instability will be important. So in conclusion, we've really learned a lot, and it's been an exciting time to be a human geneticist working in the field of HD genomic modifiers. And we're hoping to use this information generated from large cohorts of individuals to help improve the treatment of the disorder. And interrupting sequence variants, although they're rare, they're important for certain populations and subpopulations, such as symptomatic RP carriers. And as we saw earlier as well, that like the majority of African individuals actually carry non-canonical non-canonical interrupting sequence variants in the HD cohort. So it's definitely something that we can't exclude, and current diagnostic tests do not capture them, so we need to think of new approaches. So at this time, I'd like to just acknowledge the head and lab for the past research, and then my current lab as well as the conference organizers for inviting me for this presentation. Thank you.
Great. Thanks a lot, Galen, for that really nice talk. I think we have a question up front here from Elena.
Yeah, can I ask you, thanks for your talk. Maybe you said that, but I missed the point. In your loss of interruption data, in which you say that there is no somatic instability, so what is the CAG size in those samples?
In those samples, we included like individuals. We have like individuals from non-pathogenic all the way to fully penetrant individuals, and we see like with the symptomatic individuals, it's from the RP range and up. But it's a variety of CAG sizes that we use to calculate the somatic expansion scores.
Okay, but what is the highest CAG?
The highest, I think it would probably be like around 50, I think.
Great. I think we have time for one more question. I think we've got one in the back there.
Yeah, so thanks for a beautiful talk. I have a more general question, so maybe also for the previous speaker. So we heard a few times that somatic instability is measured in whole blood. But blood is, of course, not a uniform tissue and consists of many different cell types, lymphocytes, macrophages, eosinophils, etc. So what I wondered is whether somebody has looked into somatic instability in different blood cell types, and would it be worth looking into that?
Yeah, it's definitely like the cell type specificity in these repeat of somatic instability is really important. And yeah, I wouldn't be surprised if different blood cell types have different levels of somatic repeat instability, but yeah, I'm not sure of a study that's done it. But the new approaches as well that and I think one of the CHDI presentations from previously was showing that you can, there are now assays which you can look at the single cell, they've developed assays to look at the single cell level and capture gene expression to capture these like somatic instability at the cell type level. And I think we're going to learn more and more in the coming years.
And one very last question from Sarah.
Yeah, this is a quick one because it's a kind of conceptual question.
Yes.
So very nice talk, Galen.
Thanks.
So I completely agree with you about, I think it was your slide 2 saying that drug trials that are supported by genetic data are much more likely to be successful. Completely agree with you. Except in Alzheimer's disease.
Yeah.
So there is amazing evidence for APP and A-beta.
Yes.
Like very powerful genetic evidence.
Yes.
But pretty spectacular failures of 20 plus years of targeting many different aspects of different species. So I was just interested, it's a kind of bigger question.
Yeah.
It's obviously not always perfect, or
Exactly.
So I just wanted your thoughts on
Yeah,
that, because
well,
it's a kind of. I think that's why companies are still obsessing
Yes.
about APP and A beta, because of
Yeah.
the genetics.
Yeah.
And it's just what you think there.
Yeah, so that's a really good question, and like Alzheimer's disease is definitely something where there's been a lot of controversy about these drug trials and drug failures. But I think if you look at GWAS as a whole, I'm thinking now of something like schizophrenia, where we look at like now we've got like 100 hits, and it took a while to show theoretically that DRD2 was one of those hits, and we know that all antipsychotics target DRD2 receptor. So I think even if like some of the top hits fail, it's giving you more targets to look at. So it's not a perfect technique, but it helps. And what we also show, like I think it's also been shown, and like I've had reviewers on my grants before, when I mention that, they say, oh, GWAS effect size of variants are so small, it's not going to really do much. But there are other studies that show that the influence of therapeutic targeting those genes can be higher than the effect size of individual genetic variants. And when we look at things like FAN1's a great example about how we've seen this whole spectrum of variants based on allele frequencies and larger effect size for the more rare deleterious variants. So, yeah, thanks.
Great. I just wanted to thank again all the speakers today, and thank the audience for all the really good questions. And I think that'll be it for us. So enjoy lunch and the posters.
Thank you. Thank you.